Khalid.
Good morning, everybody.
Good morning.
Sorry for joining a bit late. It's a bit early over here.
Yeah, that's fine. Yeah. We were expecting that.
Yeah. Okay, so our next speaker, since we are a little over time, so I'm going to just jump to the next talk, is Suhita Nadkarni from Indian Institute of Science Education and Research in Pune. So Suhita is a very long-term friend and lab fellow. When I joined Peter Young Lab in Ohio University as a first-year graduate student, Suhita was almost on her way out.
That's not true, Ghaneem. I'm not that much older than you. Anyway.
So,
like 1 year or two.
she was in her third year, so okay. And so she, I would see actually when I first met her, and then the weeks after that, I would see her, and she was kind of this chill person, and I was like, and she seemed like almost that she doesn't care. And then she would come up with these really nice papers. Her first paper, which came out in Physical Review Letters and got a lot of attention and news in the science media. And then, I got to know her, and my first impression was, well, she does care about science a lot and is really passionate about it. So we had some, good couple of years there, and after graduation, she went to UCSD for her postdoc at the Theoretical Biological Physics Center, and then the Salk Institute, and spent significant time there and produce some really nice detailed models of calcium signaling and synaptic function. And now, after establishing her lab, which were at Pune, she has a really nice big group of people working with her. She comes out with really nice influential papers, really interesting. So I kind of from time to time just go check her website to see what she is up to, and each time I find something really interesting and kind of now share it with my students, and they enjoy her work the same way I did when I was student and a postdoc. So it's a great pleasure to have her tell us about her new work, her recent work. With that, I'll hand it over to Suhita.
Thank you, Ghaneem, for a very generous introduction. I must say, you 15 years and you haven't changed much. So can't say the same about me. And I'm really thrilled to be part of this. So thanks, Ghaneem, Atta, and Shahid for this opportunity. I just wish it wasn't online. I wish I was there in Lahore, but hopefully soon. It's definitely on my list of things to do and eat all the awesome food from Lahore, but someday soon. And thanks Padmini for setting me up so well. I almost stole my thunder, but we'll see. As Ghaneem said, I am a neurobiologist, a computational neurobiologist. I use techniques, computational techniques and techniques from physics to answer questions to understand the brain better. And this is sort of what I offer as a justification for studying the brain, right? So there's a quote from Hippocrates that goes along something like this. And men ought to know that from nothing else, but from the brain come joys, delights, laughter, and sports, and sorrows, and griefs, and despondency, and lamentations, and so on and so forth, right? So that's my excuse for doing what I do. I typically forget, I'm so sort of excited by the end of every talk that I forget to thank the people who did the work. So I would like to put this up front. The work I'm going to talk about today was primarily done by Gaurang, who's a postdoc, and Nishant Singh, who's a grad student. And this is sort of a snapshot of the lab at some point in time. I think people have moved on. Should thank my institute, ISER Pune, and the funding agency, Wellcome Trust India Alliance. So very often, brain is described as in a hierarchical structure. Having said that, this is an oversimplification. We know very well that it's intimately intimately interwoven enterprise. I think neurological disorders are a good example of how intimately these hierarchical levels are connected. So typically what happens at the level of molecules or synapses has a far-reaching consequence at the level of behavior. I work at the level of synapses, sometimes neurons, sometimes networks, depending on the questions that we have taken on to address. So this is sort of our general approach. We devise very detailed biophysical models, and we do what we call in silico experiments using these models. And given sort of our strategy to build detailed biophysical models, I guess calibration and validation of these models becomes a very important part of this modeling or scientific pipeline. And then what I'd like to argue is one of the most important contributions of modeling or computational modeling is postdiction, which is basically making sense of extant data or data that might appear at odds at each other. So I hope I'm going to convince you that postdiction is a very valuable contribution of a computational model, especially a computational model that tries to understand the brain. And given our approach is somewhat reductionist, and this we typically deal with very high dimensional space, I think a very challenging question is, of course, the importance of relevance of each degree of freedom towards function, right? So with sort of that backdrop, I think Padmini also alluded to this, that this is a very nice starting point and sort of sets us thinking, right? So our brain is about 3 pounds or 1.5 kilograms, uses up 25% of our energy, and this is by far obviously not the largest brain. For example, an elephant's brain is 5 kg and a whale's is 9 kg, but we do have the largest number of neurons up for our body weight. So that's about 80 to 85 billion neurons. And so we also have the largest cerebral cortex, which is supposed to be the seed of abstract and logical reasoning. So the question is, of course, what allows us to have this really expensive machinery? And an interesting answer to that is actually is our ability to cook. Interesting side story, but I'll tell you a bit about this. But think about it, right? A primate that weighs about 25 kilograms can afford about 50 billion neurons, and it has to graze for about 7 to 9 hours, right? Or for example, gorilla can only afford about 30 billion neurons for its large-sized body. So 1.5 million years ago, maybe a little bit more, cooking is what allows us to sort of have pre-digested food that is easy to chew, and so you get a bigger yield of energy for the time consumed, and so it sort of leaves us time to, I guess, write things like poetry, right? Where does all this energy go? As again, Padmini sort of set me up very nicely. A lot of it goes in synaptic transmission, right? In the exchange of ions back and forth, and so on and so forth. So I'm going to talk to you about synaptic transmission, and so out of the total amount of energy spent, I think close to 60% goes in synaptic transmission. And just to go over quickly the chronology of events that constitute what we call a successful transmission of signal is something like this, right? So Here is the point at which two neurons are making a contact. This is the presynaptic terminal, the postsynaptic membrane. Typically, an action potential comes in that sort of leads to these bunch of calcium channels, which are voltage dependent, VDCCs. And because of the action potential, the change in membrane potential opens them up, leads to bunch of calcium coming in. And if this calcium binds to the vesicle release machinery that is holding these pouches of chemicals, the vesicles, then these vesicles fuse with the membrane and chemicals are released. These chemicals go and bind to the postsynaptic receptors, and a successful signal, postsynaptic signal is created, right? So this is what you would call successful synaptic transmission. Now, the kind of synapse I am interested in, the CA3, CA1 synapse is in the hippocampus, which is supposed to be the center for learning, critical for learning. In fact, this particular synapse has been associated with experience-dependent learning, spatial navigation. Now, what is interesting is that this particular synapse is very low fidelity, is highly unreliable. So only about 20% of the time that an action potential comes in, a vesicle is released, right? So you sort of want to ask, is this a bug or an attribute? I mean, is this what you want a synapse that is associated with learning to be so low in fidelity or so unreliable, or is this somehow designed around wanting to save energy? Another classifying attribute of the synapse is that this particular low reliability or low release rate of the vesicle actually is something that can be tuned up. So this rate can go up, and this is what we call facilitation, and it's sort of classified in this broad name called short-term plasticity. So within few milliseconds, tens of milliseconds, you can ramp up this release rate. That's something to remember. Synapse is no longer just a pre and post, and this is just for so that I make sure that I give you a complete story. A synapse more recently, as of 15 years ago, since 15 years, is identified as something with a third part, the process of an astrocyte. So especially in the hippocampus, it's most synapses are enveloped by this cell called astrocytes, which is not electrically excitable, but has calcium-driven excitability. But again, that's a story for another day. So of course, Ghanim, I wonder if you remember this cartoon. This cartoon was made by Peter several years ago. And if everybody had to pay $1 to Peter for the times that which they use this, I think it would be a millionaire. But unfortunately, that hasn't happened. So this is what a physicist thinks synapse looks like. And this is really what a synapse is. And this is a presynaptic gluton. It's gunk of proteins, several ion channels, receptors, machinery associated with vesicle release and vesicle recycling, several proteins associated with synaptic signaling, secondary signaling, all operating at multiple time scales. And this is, of course, a canonical synapse reconstructed from super resolution microscopy. Now, synaptic morphologies differ across not just phylogeny, but across brain areas. So, as it turns out, understanding structure-function relationship at the level of synapses has been one of the most enduring questions in neuroscience. What I'm going to talk about today are other fundamental constraints, which is basically information transmission and energy consumption that go into neural organization, and how we can use these to understand structure-function relationship of a CA3, CA1 synapse in the hippocampus. So this is hippocampus, looks like a seahorse. And these are what the synapses inside. Can I just want to move this on the, okay, there you go. So these are about 500 synapses reconstructed from CA3, CA1 region. The entire thing is about 5 cross 5 cross 5. So all this, spatial complexity that both Padmini and I are very excited about fits into a single size of a single red blood cell. What I'm going to show you is a realistic sort of chronology of events that ultimately leads to a vesicle release. Given that this is a low fidelity, highly unreliable synapse, I had to run these simulations multiple times before we could see a release. So I have actually made all the presynaptic boutons disappear. What you see here is just single presynaptic bouton in green. And then as I run this movie, you'll see that there's some basal activity of calcium. And at some point, there'll be an action potential that arrives, and then there'll be sudden flux of calcium from outside to the inside. And so these yellow tiny balls is the basal level calcium. This volume basically implies that there are about only 15 to 20 calcium ions moving around. See, now this huge bunch of calcium. At some point, there will be a vesicle release. There you go. So that's a bunch of glutamate being released postsynaptically. Secondary signals starting off in the postsynaptic neuron. So this movie uses M-cell, and again was done in collaboration with Tom Bartol. To give you sort of, since I was talking about diversity of synapses across brain areas, let me show you another synapse. This is the neuromuscular junction. You can see that there's these white balls are actually bunch of vesicles lined up very nicely on the neuronal membrane. Now, in this case, it's a diametrically opposite synaptic design. Here's, in fact, a redundant amount of neurotransmitter is released in response to a single action potential. And you sort of think that this is what you want. This is a neuromuscular junction. When you want your arm to move, it should move, right? And that's exactly what seems to be the design principle here. So here's sort of what it looks like. There you go. Bunch of, again, this sort of, this is from realistic synapse, NMJ, neuromuscular junction. Bunch of vesicles lined up, receptors sort of optimally placed on the muscle membrane. And eventually you'll see just a huge amount of like a meteor shower. And that's all this acetylcholine that's released in response to a single action potential. So just want to emphasize the whole diversity of synaptic design, which again suggests a very strong structure function relationship. Let me just let you see this whole beautiful meteor shower before I go to the next slide. Okay, there you go. And these are tiny acetylcholine molecules dropping down. They'll bind to acetylcholine receptors. Acetylcholine esterases will quickly break it down so that your muscles don't continue to have tremors. So this has to be temporarily really precise signal that has to be triggered. Back to the synapse of interest, the CA3, CA1 synapse, particularly CA3 presynaptic terminal. What you see here, remember I mentioned that this particular synapse is associated with experience-dependent learning and spatial navigation. So what I mean by that is a subset of these neurons uniquely fire action potentials in response to an animal being in a specific location. So what you see here in green is the trajectory of an animal running around. And every time it crosses this yellow circle, a particular neuron, say this neuron, is showing up activity. Okay, and that's why these cells are called place cells. And this is sort of a typical what information carrying input signal looks like in these cells. So what is it? It's a bunch of action potentials, what we call bursts, and each epoch can last from about half a second to second. And each of these epochs, which is basically the rate at which the animal is visiting this yellow circle, it can be anywhere from 5 to 20 seconds. The frequency at which the firing takes place in each of these epochs is measured to be between 5 and 60 hertz. So this is what we think of as an information carrying signal. This is what is given as an input to a CA3 neuron. And so we define information in this input signal as a standard Shannon measure. Okay, so now remember what I told you before, that this is a highly unreliable synapse. So for example, every single time there's an action potential, you don't have a release. So this NR, which is the number of times, number of releases, is sort of a function.
Sorry to interrupt. Sorry, Suhita,
Yeah.
sorry to interrupt. This is Sriram. What is this phi that you are summing over to define the Shannon entropy? I just don't know. What is the state variable that you're talking about?
So this is basically, you define this as, oh, okay, sorry. So this is the signal. Phi is the signal. And the rate at which, the probability of phi is the rate at which the animal is traversing the particular spot.
So I'm still confused. I mean, when you sum over phi, what are you summing over?
So this basically the phi that you're inputting is number of action potentials. The time over which you are summing is typically order of tenths of seconds.
Nice. Okay.
And
okay,
this, yeah, this is the.
Okay. Okay. Thanks.
Yeah. And so the postsynaptic response is basically number of neurotransmitter releases, which is sort of a function of the frequency at which the presynaptic signal is coming in, right? And so the fidelity of information transmission is the entropy of this synaptic release minus the entropy of the noise, okay? Okay, so how do we define efficiency or the cost? Well, we define energy consumption as basically the rate of vesicle release, which is sort of a proxy, but a reasonable proxy, which has been sort of consistently used by several people. And so the cost is defined as the energy used, which is vesicle release divided by the information. Okay. What I should tell you a little bit about is that how do we talk about the probabilities, right, of release or the postsynaptic response? So remember I mentioned that the postsynaptic release probability is really, really small. So this is the PV is defined as release probability of a single vesicle. PS is the release probability of the entire synapse. So the probability that a vesicle is released from the entire synapse is basically 1 minus none of the vesicles released, where NRRP is the pool of vesicle releases or a pool of vesicles available, which tends to be really small. So this tends to be sort of the order of no more than 5 to 7 vesicles. And so how does the intrinsic vesicle release probability change? Well, as I said, that one of the defining attributes of this synapse is that its ability to increase its rate in response to ongoing activity. And so this is described by this particular parameter, alpha f, the gain parameter. 1 minus alpha f, 1 minus PV makes sure that the probabilities never go beyond 1. the delta function, basically every single time there's a action potential, the PV can go up. And it can go back to its resting state, PV0 over time tau f, if there is no activity. So, and this tau f is of the order of about 150, 140 milliseconds. So any activity that is shorter than 140 milliseconds can facilitate the synapse quite drastically. So this is sort of what.
Could you please switch to full screen mode so we can see?
Sure, yeah. Oh yeah. Sorry, yeah. Oh, so there you go. So yeah. So this is sort of the big picture of the synaptic transmission. You start off with some NRRP number of vesicles, which are of the order of 5 to 7. And what happens is that if there is activity, the synapse, the release rate gets facilitated by a factor of alpha f. If there's a release event, then of course the number of vesicles that are available is reduced by that number, and this replenishment can happen over much longer time scale. So there can be a depletion. What does this mean, right? Where does this take us to in terms of short-term plasticity? So what I show you here on the x-axis is release probabilities, and on the y-axis is what we call a paired pulse ratio, which is basically a measure of short-term plasticity. It's basically a ratio of release probability to the second pulse divided by the release probability of the first pulse. So anything more than 1, the synapse is facilitated. Anything less than 1, the synapse is depressed. So what you see here is that the synapse seems to have the most amount of facilitation at low release probability, or alternately, facilitation drops down with release probability. Now, of course, the trivial way to understand this is if the synapse is already operating at high release probability, since this is a ratio, there's less room to move up. The other reason why this follows this exponentially decaying dependence is because of the limited vesicle release resource. So once you deplete the vesicle release, or once you release a vesicle, the vesicle resource is depleted, and therefore your PV, your PS, the probability of the entire synapse is actually proportionately then going down. And again, on what this shows is that this sort of behavior is consistently true across different time periods between the two pulses, right? So for this one was for 40 milliseconds between pulse 1 and pulse 2, and this is over a wide range of ISIs or inter-stimulus intervals. Separately, you also see very clearly here that that facilitation is highest when synaptic release probabilities are lowest. Okay. What you see here is that is response to a train of stimuli. Again, with stimulus coming at this rapid rate, you can deplete your vesicle resources, and you sort of see for a low release probability synapse, the synapse starts to facilitate, the green one, and then at some point starts to depress. Okay, so this is sort of the full chronology of events. When you turn on the full model, you sort of sample. So Sriram, maybe this would be somewhat clearer from this visual, that you're sampling from this uniform distribution. The distribution is between 5 to 60 hertz. This is what we measure from experiments, or not we measure, but it's what's measured from the experiments. And these epoch of spikes are basically Poissonian with means from this uniform distribution. This leads to some dynamic changes in release probabilities, the PS and PV. and some vesicle release events that we bin in appropriate bin sizes. Okay. And so we do various things, and which I probably don't have time to show, is that we of course vary these bin sizes, we vary, but keep the bin sizes fixed, and vary the frequency at which each epoch fires. So all these consistently show this very robust result. So now I'm showing you normalized information transmission on the y-axis, and vesicle release probability on the x-axis. What is easy to see is that information goes up with basal release probability. The black line is for when the synapse does not show this short-term facilitation, or this factor alpha F that I described in detail is 0. And as you increase alpha, your facilitation seems to, or your information seems to go up. Okay. What is interesting is at alpha values about 0.03, the synapse seems to be operating at maximum or close to maximum information transmission. Not only that, it seems to be robust or insensitive to the basal release probability. So the maximum transmission of information seems to be robust across 4 orders of magnitude of the basal release probability, right? So the interesting sort of insight or magic about this is that, as it turns out, this alpha F is the, as 0.03 is the number that pops out when we map our simple phenomenological model to diverse data on this particular synapse. The nice thing about this synapse is that it's been studied as a prototype for learning and plasticity. So there's huge amount of very rich data sets that can be used to bootstrap some of these parameters. So again, at this particular value, 0.03, the synapse seems to be operating at maximal information transmission rates. So biological synapse, in summary, does this over a range, 5 orders of 4 orders of release probabilities. So that was interesting. What about energy use, right? So I've just simple vesicle release rate, and predictably, as release probability goes up, energy use also goes up, which also is seen in the cost. So on the right hand, the data that I show you is the cost that I showed you the formulation for, right? Which is basically energy used by relative information. So again, what seems to be happening here is that a synapse without any facilitation seems to be the most efficient synapse, or operates at the lowest cost, which is, of course, at the cost of losing a lot of information. So even though you're operating cheaply, you lose a lot of information. However, as you increase alpha, costs go up drastically, albeit you gain information. So of course the question is, are biological synapses energetically inefficient? Does it not care about energy constraints at all? The answer seems to be somewhat nuanced. So this is again now data in comparison to biologically relevant value of alpha F, the facilitation parameter. So so this is sort of percentage change. Blue is information, orange is energy. What you see is that below biologically realistic facilitation values, you of course operate at really low energy, but also lose a lot of energy. Whereas if you keep increasing alpha, at some point above the biologically sort of relevant value of alpha, you don't seem to gain much in terms of information, but your energy costs seems to go up drastically, right? So this is sort of the cartoon of what I just showed you in data. So on the x-axis is the energy cost, on the y-axis is the information capacity. And as you increase facilitation, you of course expend more energy, but you gain information. But beyond a certain point, you're not gaining in terms of information, but you keep expending more energy. And I guess the magic part of it is that biological synapses seem to be operating in this very nice sweet spot where it maximizes information but penalizes excessive energy cost. Now, even though I say it's magic, I think what I want to emphasize is actually it's really not, right? The reason we get this really nice result is because we invested in finding out what was the energy bearing information signal, right? In this case, what do the place cell firing looks like? What are the temporal scale associated with the firing? And that sort of married well with the facilitation time scale. So when I showed you the data on low release probability synapses facilitating really quickly, given that you have an epoch of about half a second, it doesn't matter what basal level release probabilities are, you're ultimately quickly catching up and transmitting all this information, right? So magic or no magic, it's up to you to decide. But with this, I'll sort of end my talk. To summarize, information is maximized in an energetically cost-effective manner. Padmini spoke about how information is encoded in the in spines. Specifically in the C3-CA1 synapses, information seems to also be encoded or stored in the presynaptic terminal. And one of the ways in which this information is stored is by way of long-lasting change in presynaptic strengths. So even as ongoing experiences are stored as changes in release probability, what we see here is that because of short-term facilitation, these synapses are still able to maximize information transmission, right? So that's quite interesting. And again, to sort of emphasize that this general principle came out because of an understanding of the local environment of the synapse. So this principle might not hold at neuromuscular junction. Other large synapses in the hippocampus, for example, the DG or the calyx of Held, which is a synapse in the auditory cortex, and so on and so forth. So I think design principles must be addressed in a local context. There are several functional roles associated with short-term plasticity. Working memories associated with the short-term plasticity, short-term plasticity and facilitation is also associated with synapse being able to act as a high-pass filter. This is sort of a novel role that we assign to short-term plasticity, which is that it maximizes information transmission. And I guess that's it. I am happy to take questions.
So, Suhita, I have a question.
Yeah.
So you've shown very nicely this relationship between, information transfer and, energy utilization. And so if you have a kind of a two-dimensional phase diagram where information is on one axis, and which is essentially what you've got there. But now I'm wondering, is there possibly a third dimension which could be the frequency of the stimulation, right? I mean, like,
Yes,
or do you only need to worry about this trade-off between energy and information?
No,
or
you're right.
is there a timing issue also
There
involved
is.
in here?
I think you're exactly right. The only reason we get this elegant sort of relationship is because we specifically relied on certain statistics of the input signal, which is relevant to the synapse. The reason we get maximal information transmission is because of the width of the epoch, the frequency within the epoch, and the time between each epoch. So the time between each epoch allows the limited vesicle resource to be refilled. So details matter. And that's, even though it's almost not surprising, it's still very interesting. That optimality principle has sort of evolved in a very local context. So if I had to give a regular train of 30 hertz, I would get depletion, and my results will not hold up. And I think that's actually interesting rather than not interesting.
Yeah, no, I agree. Yeah, I mean, absolutely. And then
to higher governing pressures,
yes,
because of which you arrive at a particular design. And that it is robust to a range of basal release probability is also interesting, because you do want to have this heterogeneity of synaptic designs. This is where your information is stored.
Yeah, and I think if you get to that, that when your alpha is 0.3, I mean, you're really being, you have a wide dynamic range, right? 5 orders of magnitude is not bad. I mean, so that pretty sort of
what
gets
I did
you where you
show
want to
you
go.
was one of the things, a fallout of this particular thing, or an additional sort of advantage of facilitation, is that it also makes synapses more reliable. And we did a separate calculation for that, which I didn't have time to show. But I think the reliability also comes from marriage of timescales.
Could I ask a question, Suhita?
Yeah, please.
So I'm wondering, so you're measuring information transmission by vesicle release in the presynaptic region. But ultimately, information transmission is the response you get in the postsynaptic region.
Basic off-the-shelf Shannon, yeah, off-the-shelf, yeah.
Right. So I'm wondering, is there, I doubt it's a linear correlation, but have people done experiments where they actually looked at the correlation between vesicle release presynaptically and then information transmission postsynaptically, maybe in the form of the calcium transient and the postsynaptic region? Because ultimately, in my mind, that's information transmission. It's not just vesicle release, because there's a lot of integration in the synapse, how the information is transmitted. And if you're going to generate memories, you have to generate a signal in the postsynaptic region, ultimately.
I think you are correct. So this is at sort of the first level of synaptic transmission, that at least the postsynaptic side has registered the event. A release necessarily implies that a postsynaptic membrane has registered the event. Whether what that event goes towards postsynaptically is a more complicated story. And probably has to be dealt with on a longer time scale, right? I mean, I think all of us will agree that the role of long-term plasticity is way more complicated than just making the synapse stronger or weaker, right? And that's why you have thousands of molecular players that act on different time scales, all triggered by calcium, right? And each of this goes towards probably a very complex way in which memories can be stored. And so I think your question is hard. I think you have to, this is the simplest possible definition of transmission of information from pre to post. I think your question takes it further to, I think, defining instantiation of, I guess, I think your memory engrams, right? I mean, what goes towards actually storage, long-term storage. And that's probably a cumulative effect of release transients. I know this is not a very satisfying answer, but That's the best, I guess, one can do.
Thank you, Suhita.
Any other questions? So, Suhita, I'll ask a quick question. So, from what I understand from the formalism, you include the cause seems to be coming from the action potential mechanism, right? That information.
Sorry, say that again, Ghanim. What was it? The what comes seems to.
So the energy cost comes from the action potential generation
yes,
mechanism.
So, yeah. So even though in this paper we have used a simple proxy for energy use, we have done the ATP calculation also, and it is consistent with vesicle release. You're right. A lot of the energy consumption that goes towards synaptic transmission is just the action potential and the fluxes of ion back and forth, right? I mean, sodium going in and calcium coming out and all of that, right? Yes.
Yeah. So my question is, if you want to do this over a longer time scale, where the vesicle replenishment would occur. Because here you basically, if you look at your figures, you deplete your vesicles and there is no replenishment. So then if you do have enough time for the replenishment, does your equation?
Ghanim, the reason the definition of what we, or the way we define signal is very, very important, is because in this scenario, depletion does not play a role, because if you look at the signal, the typical time between each epoch is about few seconds. And that's plenty of time for the vesicle pool to be replenished. And because the vesicle pool is replenished, depletion in this scenario doesn't seem to play a role. Now, people have used depletion as a way, or depression as a way to conserve energy. And this has been shown in, I think, the visual cortex, where the role of depletion in optimizing information and basically lowering energy consumption and maximizing information. In this context, the way we think it's legitimate to formulate the problem, depletion doesn't seem to play a role.
Okay, so the recycling, the energy cost due to the recycling of the vesicle is taken into account in the equation.
Yes,
it is. Okay.
This is, I think, the time scale that we use for vesicle recycling is of the order of maybe 2 to 5 seconds.
Okay. Okay, that makes sense. Okay. Are there any questions from the students? If not, thanks, Suhita, for a very nice presentation.